Okay, so chapter 3, prokaryotes, right? Again, there's a clinical case study up there, so that if that helps you to study, don't forget that there's a QR code to kind of get some of that stuff there, right? Off with the basics, right? And remember we talked about the endosymbiotic theory yesterday, how that worked and what have you, right? So we talked about that. We know that the first living organisms were prokaryotes, okay? That is a theory, right? And in theories, for example, those aren't 100% proven, okay? Not 100% proven, but that's more than likely what we think is occurring, right? Does that make sense? Theories, right? It's like saying, you know, I'm pretty sure that if I jump off of, I don't know, a 50-foot building, I'm going to fall and probably break my legs and die. But I don't know that to be true, 100%. I might live, but probably I'll die. But I'm not going to try it. You see what I'm saying? Like, that's a theory. Does that make sense? It's saying, like, we know pretty much that that's going to happen, but we're not 100%. All right. So here's the breakdown, right? We talked about a little bit of classification yesterday in chapter 1, but we know that there's different domains. Domains are the main spreading of living organisms. And we know for a fact that if this was we could call this the tree of life. This is commonly called the tree of life in biology because we know that this paints the picture like a tree of where we all come from. Does that make sense to everybody, right? We know that there is one ancestor that was a prokaryote. We just don't know what that looked like or what was the very first living organism because we have no record of it. Okay, but we know from evidence that we all come from the same living organism, and then after a certain time frame, that tree starts to then separate, like branches on a tree. Does that make sense to everybody? Right? And the two branches we're discussing are prokaryotes, the one in green and the one in yellowish color, right? We'll talk about eukaryotes, that's chapter 4, but this is where we're splitting. That makes sense to everybody? Okay. That's just so to put down if we continue with the tree, this is kind of where our branch goes. See what I'm saying? And then it branches even more and more and more. Plants would be over there, animals would be over there, human would be way over there. Okay? Makes sense to everybody, right? So we classify prokaryotes in two ways. Either they're bacteria or what we call archaea. Okay? Those two, bluish, yellow. Okay? Eukaryotic cells are in the other branch. Right? Prokaryotes by definition are unicellular. Both bacteria and archaea are unicellular. Unicellular means one cell. Okay? That one cell is their one-stop shop. That's their one organism. They don't have more than one cell, just one. That's your bacteria or your archaea, whatever. Okay? And a prokaryote, remember the prokaryote, pro-kernel, right? Means that they do not have a nucleus, right? And if they don't have a nucleus, they don't have membrane-bound organelles, which means they have organelles. It's just those organelles don't have a membrane. Okay? A boundary. This also means that they don't have a nucleus, but they do have what? DNA, right? They have DNA just like everything else that's living has DNA. It's just that DNA is not held in a little bounded area that we call a nucleus. Okay? So they have organelles, it's just not in a membrane. They have DNA, it's just not in a membrane that we call a nucleus. And they're single cell. Here's your typical prokaryote. Okay? Not every prokaryote looks like this. It's just your general kind of, if I had to put down all the parts of a typical prokaryote, this is typically what you would see. Not all the time. Notice that it does have a plasma membrane, right? Just like other living organisms. It does have DNA, like other living organisms. And it does have some organelles. Does have organelles. They just are not in the membrane. Can you see that the DNA is kind of held in an area, but there's no membrane around it, there's no nucleus, right? The purple is the DNA. Now, prokaryotes have really different shapes and sizes and, you know, all of those kind of things that come with it. Because we know that there's different organisms that are prokaryotes and they have different shapes and sizes and arrangements, we can actually classify them in that way. And we're going to begin to understand that as we go along very soon, right? Because what we're going to do in lab is we actually can identify them by their shape and by their arrangement, how their lineage, how their copies are arranged in a, let's say, under a slide, right? And hopefully you saw that in a microscope. You would see, you know, you looked at your slides and you saw those under the microscope. You saw like, oh, okay, these look different. These might be different color, but they look different. So, for example, the prokaryote bacteria that are monomorphic means that they're always going to have one type of shape. Always going to be one shaped type of molecule or type of cell. Paleomorphic means that they can take on different forms. Bless you. Can take on different forms. You'll notice in this figure, this picture, you can see that they have almost the same type of shape. Can everybody see that? I mean, they kind of curl around differently, but they're kind of like a tube or rod, right? That would be. Taking a look at the size. Man, I screwed him over. Sorry. El Chapo was just sent to prison for life. And they ordered him to pay $12.6 billion to the government. Of course, we're going to get our money from them.
But the question is, will he stay in jail this time?
Well, he's going to Colorado, I think. Colorado Supermax prison is where they hold the more, like, state-of-the-art, like, top level. 12.6 billion, folks. Damn government. That's a lot of money. The government's got to get theirs, too, from that drug money. All right? 12.6 billion. That's insane. Okay, sorry about that. I was like, oh, that's great. Taking a look at their size. Sorry. Taking a look at their size. Just to note, this is micrometers, right? This is actually micrometers, and it's a really small size. You don't need to memorize the size range, right? But just to understand that they're really small, much smaller than eukaryotic organisms. Much, much smaller. Okay? When we take a look at the species Mycoplasma, Mycoplasma is some of the smallest species of prokaryotes out there. Okay? And we take a look at another species here, but that's some of the largest, and we don't need to really know that, but those are some of the largest. So they range. There's a different range in size. Most of the prokaryotic cells and their nutrients are obtained or gathered inside of their cell through a process called diffusion. Diffusion is moving molecules from one concentration to another, or taking molecules from the outside of the cell towards the inside of the cell. Okay? Since they are so small, okay, since they're so small, what we call the surface area to volume ratio is also small. And because it's also small, they take in a limited amount of nutrients. They can only gather so many nutrients because they're so small. Because of their shape, we call that surface area to volume ratio. The reason why we talk about that is we talk about the question, why don't they grow in big size? Why don't we see bacteria huge? Why don't we see a huge bacteria? Why are we never going to see a bacteria, I don't know, the size of my hand? Right? Because they can only take in a certain amount of nutrients at one time, and because of that, they say the size that they say. Does that make sense to you? That's what we mean by this. So looking at the shape, the shapes are different, and there's some terms here that you need to be aware of, right? So anything bolded here should be definitions you need to be aware of, right? Those are all shapes, various shapes, and there's actually more to this list, but those are the main ones where most bacteria, for example, or prokaryotes are going to fall in. Okay? They're going to be bacillus, which means they form a rod. Their shape of the cell is a rod. Okay? Or they can be cocci, which means they can be circular shape or sphere shape. Shouldn't say circular, but it's more sphere, like a ball, right? Vibrio means comma shaped. Some organisms can be stellar, which means they're a star shaped kind of formation. Coccobacilli, ovoid or oval shape, they're kind of between a rod and a ball. And spirochetes are really cool because they look like a corkscrew. And they commonly get confused with worms. People think they're parasitic worms. They're actually bacteria. They have this corkscrew shape. Really interesting. Now, you need to be aware of the arrangements as well. Okay? So what do we mean by arrangements? When prokaryotes reproduce, okay, under cell division, because of cell division, that's how they reproduce. They divide their cells. So 1 becomes 2, right? And those 2 become 4. Those 4 become 8, 8, 16, 16, 32, 32, 64, 64, 128. And they do that, right? When they become 1 turns into 2 and 2, 4. When they do that, they form an arrangement or a pattern, right? That makes sense? Pattern? And these are the patterns that some of them can turn into. Now, the reason why we study these is because we know already, because of research, many scientists already looked at this, that we know for certain that if we look at a species, we know that species is going to have a shape, a size, and an arrangement. Right? Does that make sense? So if you go under a microscope and you say, I'm going to pull blood from a patient because they're sick, and I go run a slide like you guys saw, right? Microscope slide, and we look underneath, and I see, oh, it's a rod, and it is in pairs, let's say, Diplobacilli. Oh, I know that all of these species are those types of arrangement and shape. Then I can start ruling out other ones that are not. That's kind of why we see those. You see that there, right? Can you see that they're kind of almost stuck together? Because they underwent binary fission, and then they never left each other. They kind of just started forming chains. Some of them separate, and they can become by themselves, and that's where you see this. Or the top line. Does that make sense to you? Right? That's giving a quick introduction to prokaryotic cells. And the last topic we left off on was looking at binary fission and how that property works. Now, not all organisms will undergo binary fission, right? Not all prokaryotes will do this, but most will, okay? Most will. So as far as you need to know, that's really how they're reproducing for the most part for most organisms that are prokaryotic. And again, we'll talk later on about time frames and how this works as far as that is concerned, but again, this is going to be an important topic to understand, because when we talk about how bacteria grow in the human body, what type of system do they invade, one of the things we talk about is when somebody gets ill from an illness, right, from an infection that's pathogenic, it all has to do as well with pathogenic load. And what we mean by that, pathogenic load, is how many of these individual estimated number of organisms entered the body somehow, some way. That's the pathogenic load, right? And if you have a small pathogenic load, and it just depends on the organism, then your chance of infection, contamination and infection in the human body is very, very, very low. Now, on the other side, depends on the bacteria and depends on how much of that bacteria numbers you got exposed to. That could be really, really high that you might have an infection. Does that make sense to you? Right? So, yeah, things are going to be contaminated. Obviously, this afternoon when we go to lab, you're going to see that bacteria live everywhere. That's the really take home of that, one of those exercises, that bacteria live everywhere, including your body, in your body, on your body, and on surfaces. It just depends on the actual odds of it getting into your body that is pathogenic. On top of that is where we see this. But when we talk about the load, we talk about how many of these are reproducing at one time. Does that make sense, right? So yes, you might have pathogenic bacteria, I don't know, on your raw chicken that you're going to cook tonight. Yes, right? You're going to cook chicken tonight, let's say. You might have bacteria on there already. But how many of those do you have? More than likely a very, very, very small amount. Number one. And number two, can the digestive system and your immune system take care of it, which means kill it before it actually gets into like tissue that it wants to reproduce in, right? Now, if you were to take the chicken and cook it the proper way, take it out of the refrigerator, don't leave it out in room temperature too long, don't let it touch other surfaces, cook it properly for the right time and temperature, all that's good. You probably eliminated most, if not all, that bacteria anyways, right? And if you take that side versus, let's say, another scenario where you leave the chicken out for, I don't know, 2 hours on the front counter, don't cook it the right temperature, a little raw inside, and then decide to eat it or let it rest for a while and then eat it later on, well then what is happening is probably that bacteria that's in your chicken has done this many, many, many times. So the pathogenic load is what? Huge. Does that make sense? Now what you've done is ingested a lot of individual bacteria. And if those individual bacteria are bad, like salmonella, classic, coupled with food poisoning, food sickness, it's a bacteria, likes to live in cold temperatures. It almost pairs perfectly with like raw meats and chicken and meat, deli meats and stuff like that. Let's say it's that, then you're going to get sick. Does that make sense to you? That's why they say certain temperature or a certain type of meat, don't prep it here, take it right out of the refrigerator. There's warnings to all that because, again, this is what we know to be true. Okay? All right. So next we're going to move on to extracellular structure. So we're going to look at the outside portion of, so we've gotten a little bit of introduction. Now we're going to go to the outside structure of a prokaryote, typical prokaryote. What is on the outside, one being, of course, the plasma membrane, right? All living organisms, per se, have a plasma membrane, right? All living organisms do. That's just the truth of the matter, right? Doesn't matter if you're talking about a plant or an animal or even bacteria and archaea, they all have plasma membranes, okay? So what is a plasma membrane? Basically, you can think of very simply as a barrier, a border, that separates the outside environment from the inside environment of the cell. So it makes a cell what a cell is, right? That it's its own little containment unit, right? And it, again, is a barrier between outside and inside, okay? The outside things, structures, chemicals, it can be molecules, whatever, are considered extracellular structures. Extra means out. Of course, structures, objects, chemicals, whatever, extracellular. And stuff inside is intracellular structures, right? Inside the cell versus outside the cell. What's the border in between? The plasma membrane. Very simply. Now, most prokaryotes, not all, because I always say most, not all, because there's going to be that kind of thing. There's not a always for a lot of this, right? Most prokaryotes have a cell wall, right? Which is not the plasma membrane. Okay, so some students get confused. They think cell wall and plasma membrane mean the same thing, and that is not true. The cell wall for most prokaryotes that have it will lie on the outside of the plasma membrane. So plasma membrane, right? Plasma membrane, and then can't get that. There it is. Let's say that is the cell wall, right? So two barriers basically that a prokaryote contains. Now, why would this be true? Why would this be something that, let's say, prokaryotes have, but eukaryotes probably don't have? So what I'm saying is cell walls exist mostly in prokaryotes. We do have some in eukaryotes, but not many. And you might think to yourself, why is that the case, that it would have an extra kind of barrier? Well, the reason why is because these exist in the environment. So the environment is a rough kind of, for your own self, only the strong survive kind of thing. And this helps you survive better, basically, in these environments. Okay? Again, it's just one cell. That's your one organism, one everything. So we know that a plasma membrane is made of phospholipids, right? They highlight for you a phospholipid there. They highlight a phospholipid for you there, right? It's one little phospholipid. That's the little, looks like that, right? Head and tail, okay? And it's a phospholipid bilayer, right? It means two layers. There's layer number 1 and layer number 2 right there, right? Layers where the tails point inside of themselves. The head is what we call hydrophilic. Okay, it's a hydrophilic chemical or molecule portion of the phospholipid. And the fatty acids are hydrophobic, which means water hating. Okay, where most of this outside the cell is water, and because it is water, it's designed this way so that water doesn't just flood, all the time inside and cause the cell to change shape and blow up and stuff like that. So what it's designed for is that it prevents, for example, water and water-like chemicals from entering through the phospholipid. That makes sense? Now the little head is water-loving, so water and water-like chemicals can interact with it. But if it goes past that head and hits the tails, it'll stay behind. It'll stay in the outside. It's only when the cell wants to bring something in or in out, including water, it can do that, but not through the phospholipid bilayer. Okay? Now, what you also have are proteins, and these purple things are represented by proteins, and we'll talk about those as well. And then you also have carbohydrates that exist on the outside of the cell, and there's one of them there, kind of looks like a little, piece of grass or a branch of a tree or whatever. Okay, and we'll talk about all those as well. As it mentions, it's a selective barrier, so it only will select what to come in and come out, and the way it selects, or the way it uses to select, is these proteins, basically. Not many things can go through the phospholipid bilayer, as I mentioned, so that's why it makes it selective. Okay? Okay? So talking about proteins, discussing proteins, because we looked at proteins really quick in the figure there, but looking at proteins, in fact, half of the mass of the plasma membrane is proteins. So half of it is pure protein on the plasma membrane, not the entire cell, but plasma membrane. And so what do they do, the ones that exist in the plasma membrane? They can act as transporters. They can transport stuff in and out or across the cell. Okay, and that goes to the those are the proteins in the plasma membrane. They can anchor stuff to the plasma membrane, anchor the whole cell to other things. They can be receptors. They can receive information from the outside. We call this a stimulation, right? Receive stuff from the outside the cell, right? So they're receptors. That's what we mean by receptors. And they can act as enzymes. I'm sorry.
No, I was thinking that receptors are like receiving like signals and stuff.
Yes, yeah. Signal reception. Chemical reception can be a signal. Any type of stimulus. Can be another cell, right? So another cell can touch, like another bacteria can touch it, and then that's how they'll, receive that information. And enzymes, right? Enzymes are catalysts, right? They can do something with a chemical reaction. Enzymes will usually break down or build up something, right? So that's what they're designed to do. And again, I mentioned that it's selectively permeable. It's a selective barrier. That's what I just, mentioned. But it's selectively permeable. What does permeability mean? The capability of something passing through, right? That's permeability. Okay? It's selective. Doesn't mean they just don't open the door and let everything in. Okay? So these cells will select what to bring in and what to send out at any given time. And that's going to change, right? Depending on environmental conditions, internal cell conditions, things will go back and forth depending on that. Okay? So what do you mean by things, right? Chemicals that are gas, a chemical like water, molecule like water. Small, non-charged, doesn't have a charged substance. Charge means, does it carry an electron or is it not a carrying electron? Is it an ion, right? Basically. Now it mentions to you here that these things can go back and forth through the cell wall. Through the plasma membrane. And they can. So this is an edit from what I just mentioned about water, right? It depends on how big the molecule is of gas, for example, can pass through, and if it's non-charged as well. If the substance is small and non-charged, it can pass through. And in fact, it was not forthright at the beginning to tell you that water can pass through, but in very, very, very small amounts. Almost so much small that we don't really count it as passing through per se. Although it does, we have to technically say that water can pass through. It just takes a really long time, and it's a very small amount. So much so that the cell is not going to wait for that water to come in. If it needs water, it's going to use another method to bring in water. That's not going through the plasma membrane. That's what we mean by that. Now, big ions, larger polar substances require a transporter, which is a protein, obviously, to enter and exit the cell, to go back and forth. Okay? Most things are going to be probably this versus the other stuff, right? And if it's a large amount of water, for example, a large amount of water is going to have to use this other method of bringing in through the plasma membrane. We talk about the lipid bilayer as a fluid mosaic. You've heard this probably before if you've taken a biology class. Same method, same thought process. In the fact that nothing, including the phospholipids that make up the plasma membrane, are static, which means they don't stand still like this. So if this is my fingers, my hands are the plasma membrane. It doesn't mean that if this is a phosphol or this is a protein, it stays right here. In fact, in reality, and we've seen this in video, everything moves, right? Including all the phospholipids. They all are moving, and the proteins are moving, and the carbohydrates are moving. Everything that's a plasma membrane moves around. We call that a fluid mosaic. A mosaic is a pattern, right? That's what a mosaic is. But we call it fluid because it's all moving. including the phospholipids. The reason why we do that is because the proteins can relocate, right, where they're most needed. So let's say a organism like a bacteria is in, I don't know, a pond, okay, a pond or a river, and it's there hanging out, doesn't kind of just is moving along, and all of a sudden it runs into a bunch of nutrients on one side of the phospholipid bilayer, the plasma membrane. It will sense that and then move all of the proteins from this other end to the other side to receive all those nutrients if it needs it. That's what we mean by relocating all these proteins. Now, since it's a fluid mosaic, it is fluid, it does move, it does have this flexibility, it does have this property, right? And we know that this fluidity that we call fluidity is dictated by several things. Several things out there in the environment can actually influence how fluid or how not fluid it is, right? One is temperature. And we know that's an environmental condition, right? We have to, every living organism has to deal with temperature in some form or fashion. Humans as well, right? We've got a system set up to deal with temperature changes. Well, so do living organisms like prokaryotes, bacteria, archaea. But we know that temperature will affect their plasma membrane, and that warmer temperatures makes the plasma membrane more fluid, because remember, it's lipid based, right? A lot of it's lipid, so it's going to be more fluid. And colder temperatures reduces the fluidity, makes it harder. Is it not as fluid? Okay, we see this property for sure in lipids. Let's say if you go to your house and if you had butter, not margarine, because it's something else. It's like a Frankenstein we made in food, right? Margarine, by the way. Okay, I'm not a fan of margarine. I'm a fan of butter.
Do they taste differently?
Totally different. Totally different.
It doesn't have a taste.
There's like the country crock shit, and then there's like real, like almost like from the cow butter. It's totally different. Totally different, yeah. But if you take butter and you take it out of the refrigerator and you leave it on your counter, depends on how hot your house is, your kitchen, but that thing starts to soften up, yeah? We've all probably seen this before, right? Softens up. Temperature is affecting the lipids and their shape, basically. And because of that, they kind of loosen up, and because of that, this butter becomes really, really soft. And then let's say you would take that experiment and then you would throw it, I don't know, in the freezer for 5 minutes. Guess what happens to the butter? Rock hard, right? Really, really, really hard. Well, that's just a test to test, in fact, that temperature is affecting the lipids. And why do I mention lipids? Because the plasma membrane and the phospholipid bilayer, most of that's lipid based. Proteins also kind of harden up and loosen a little bit too in temperature. Okay, fatty acid content. Now, talking about lipids and talking about the tails of the phospholipids, they're going to be different depending on which organism you're discussing. And we don't have to get specific. You don't have to know what kind of fatty acid tails on the phospholipid bilayer this organism has or that organism has. But we just need to understand that certain types of fatty acids, and we have these two big types, right? Unsaturated and saturated fatty acids. Will change, right, the fluidity, basically. Okay? Unsaturated fatty acids don't pack tightly because that's what cold does. It kind of changes the shape, and then those fatty acids pack really tight. So it prevents that and improves the fluidity. Okay? The colder the temperature, it still stays fluid. Whereas saturated fatty acids do pack tightly in colder temperatures, and they become really rigid. Now, you're asking yourself, why in the hell does he want me to know unsaturated and saturated fatty acids and that there's two different types? Well, because we know for a fact that bacteria live in all different places on Earth and can survive in different places depending on the species. Well, one of the major reasons that they can do that is because their fatty acid kinds of fatty acids they have in their plasma membrane is going to differ. Does that make sense? I mentioned to you that salmonella, just really quick with that, back to that raw chicken thing, that salmonella survives really well in colder temperatures. So if I told you that, which one do you think kind of fatty acid they would have in their plasma membrane? The unsaturated. Because as it gets colder, which means they can still survive, their plasma membrane is still going to be fluid and not be really hard. Because if it gets really hard, it can crack, it can break open. And that's not what you want in a cell. You don't want a cell to break open because you're going to try to repair it, but if the hole's too big, you're dead, basically. If you're a bacteria, you're dead. That's why, it's one of the reasons why salmonella can survive in really cold temperatures. Folks, you can even freeze it and salmonella will still stay alive. That's why when they tell you you got raw meat or whatever, come to HEB, you split it up, put it in the freezer, then you take it out, let it refrigerate, and then say, okay, we're going to make dinner, and say, oh, forget about it, we'll go to, I don't know, Julio's or something like that. And I put the food back into the freezer, and then I take it out, I put it back in, or whatever I do. That's a recipe for food poisoning or an infection in your GI tract, because maybe say that meat does have salmonella, but in very small amounts. But what you've done is you've put it in a higher temperature so it grows more, and then you put it in the freezer and it stays still alive, and then you take it out and it'll grow some more. So by the time you eat it, you've got a lot of salmonella bacteria.
I'm guilty of that.
Yeah, don't do that. Now you know not to do that. You only can freeze it once. That's the rule, basically, for raw meat. Other things is a little bit different, but raw meat is one of those kind of iffies. Yeah. I would say probably breads and stuff like that is not a big deal, but the meats are. Don't, if you freeze it once, then you can't refreeze it. That's the general rule, though. Now, have I done that before? Yeah, I mean, I've never gotten sick, but you never know. You never know. Okay. As far as the length of the fatty acid, it's going to vary. Bacteria have a linear fatty acid tail, which means it's in one line. And archaea have a long branch fatty acid, which means it's a longer chain, and it looks like a little bit. So this is sometimes how we see things here as well, as far as the membrane. Here's a phospholipid in a bacteria. And again, we're going to concentrate on bacteria mainly in prokaryotes, right? We're going to concentrate on that mainly, right? And because of that, they only have bilayers. That's all we've seen so far in bacteria. They have a phospholipid bilayer. Now, archaea are a little bit different. We won't talk a lot about archaea. We will talk about it here, but we won't discuss it fully. One, because one of the major reasons is because archaea are not known to be pathogenic at all. We don't have any archaea that are pathogenic, at least not yet, which means we're discovering in the earth more and more, and then we might do that in the future, might see something in the future, but all the archaea that we know so far are non-pathogenic. Because they're non-pathogenic, and this course is for allied health, we don't really focus on that too much, okay? But if you look at the archaea phospholipids, some of them have a bilayer, like bacteria do. Some of them have what we call a monolayer, which means there's not two layers, it's just one long branch of fatty acids, just like that, with another tail like that, or another head on the other side. So they're very interesting. It's different than what you've seen in other living organisms. So they have that particular characteristic. Now look at the chemistry. So this little head and tail, or tails kind of thing, actually is chemistry, right? They're chemical. It's a chemical formula. It's a chemical molecule, right? So you've got a phosphate group here, glycerol backbone. There's an ester group there. Not need to know this, and then here's all your fatty acids down here, right? Look at the fatty acids here on the bacteria. Look at the fatty acids there on the archaea, right? What is different? They have a little kind of stick, if you will, sticking out, right? Which tells us there's a bond there, right? It's carbon bond. So that's actually at the end there'd be a carbon atom on either end like this. And every place where the lines meet, there's a carbon atom, just like this, basically. Okay? Okay, so let's talk about the cell wall. So a cell wall is very rigid. Okay, it's a rigid structure. It's a extra layer of protection. It's a survival, characteristic. Cell walls are not plasma membrane, right? They're different. Bacteria use a chemical that we call peptidoglycan, which is actually a peptide bonded to a glucose molecule, and peptides are long chains of amino acids, which is a precursor to a protein, right? It's like a protein and a carbohydrate bonded together. And bacteria use this to construct their cell wall. Or you can say their cell wall is made up of peptidoglycan. Okay? So it's carbohydrates bonded to carbohydrates, bonded to peptides or amino acids in a chain, bonded to carbohydrates, bonded, and it makes this kind of pattern that makes the cell wall the cell wall. And archaea use pseudo-peptidoglycan, which is not the actual exact same thing as bacteria, but it's very closely related. Again, we don't focus too much on archaea. Acids are found in gram-positive. Gram-negative, you don't have those. There's no tictoic acids. So if I was to ask you a question, like an essay question on the exam, like compare and contrast gram-positive and gram-negative cell walls in a prokaryote, you will be able to tell me the channel proteins that are porins on the gram-negative. You'd be able to tell me it's a thin cell wall, but it compensates for a phospholipid bilayer, that it has lipopolysaccharides, right? And that gram-positive has no outer membrane or phospholipid bilayer, has a thick cell wall, has tictoic acids. You could make a compare and contrast. Right? Okay. Again, that might be a hint for the exam. Okay, here's a table here, right, in the book that shows you the compare and contrast of gram-negative and gram-positive bacteria. So this is a good table to study if you're looking at what the difference is, right? What the similarities are, because there's similarities, obviously, and there's also differences. Look at this one. I just want to show you this down here at the bottom. Penicillin is an antibiotic. It's our most widely used antibiotic, right? I'm sure many of you already had penicillin once in your life for some kind of infection, or your children, or your whatever, right? Look at the susceptibility. That means how sensitive is it to penicillin treatment? Look at gram-negative, very low, right? Which means you can give penicillin to an infection, gram-negative infection, and it's got a very low susceptibility. It doesn't really do much to kill the bacteria. Look at the gram-positive, high susceptibility. So it works great with gram-positive bacteria. So if you have a gram-positive bacterial infection, then penicillin works great for that. Does that make sense? Now, how do you know that you have a gram-positive, gram-negative infection? If you go to a doctor, hospital, whatever. Well, one, it's the assessment, right? It's the looking at signs and symptoms, looking at the patient, medical history, where have you been, what did you do, those kind of things. Blood tests are really nice to have. Biopsy tests, taking samples of the infection and then running in the lab. Lab work is really important to understand that, but also good knowledge and good practice, right? The practicing is also really important. And I mention that because we are coming from an age right now where doctors don't have enough time, patience, energy to actually diagnose this properly, or clinicians that help out doctors diagnose this properly. And so what they do is they usually give out like one of these broad spectrum. What you mean by broad spectrum is it kind of can kill almost everything. But if you have a gram-negative infection and you give penicillin and it won't work, then you're still going to have issues there. Right? Does that make sense?
And you're just giving the gram-negative a little, like you're reinforcing it so it can become resistant to that drug. Really?
Yes, very good. Yeah, very good. It's exactly true as well. So that's another side effect that you're actually giving if it's a gram-negative infection and you're giving penicillin, it will give the potential opportunity for that bacteria to become resistant to this and to resistant to everything else. There's another weird thing that happens is that sometimes the resistance can be spread from one species to another species. And that becomes really quite an issue. Yeah. Okay, let me mention acid-fast staining. Okay, acid-fast staining. Okay, a staining technique we call acid-fast, and I believe we'll do this today if the bacteria is growing, because I grew bacteria when you guys were doing your experiments on Tuesday, and hopefully the bacteria is growing. We'll check it here in a little bit. Acid-fast staining works on acid-fast bacteria. And what they do is they detect a waxy layer that we call mycolic acid. And what this means is that acid-fast bacteria have a cell wall made up of mycolic acid. This is another kind of a chemical. And I mentioned most prokaryotes, their cell wall is made up of what? Peptidoglycans, right? Most of them. The vast majority of prokaryotes have peptidoglycan as their cell wall composition. Well, the little ones that don't have peptidoglycan have, in fact, mycolic acid. Because of that, we cannot gram stain these type of bacteria. When we gram stain them, it doesn't work, because the gram staining is only for cell walls that have peptidoglycan. Now we've invented another way to detect those bacteria that have mycolic acid in their cell wall. Does that make sense?
How would you know the difference between a gram-negative and acid-fast?
You wouldn't. You wouldn't really. So what you would have to do if you really wanted to get the full aspect of it is gram-negative, take a sample and gram stain that, and take that same sample, not the exact same one, but take another sample of the same source, and then acid-fast. What you would see, hopefully, is red-pink in that acid-fast, if it in fact is. If you see it purple over here or some other color, then you know it's not acid-fast. And if you see the gram-positive as a color, that's what you would see. Because when you apply a gram-negative staining technique to this, it doesn't hold any of the dye.
Oh, okay.
Yeah, it won't hold it at all. So the two genuses here that we see that are acid-fast are Nocardia and Mycobacterium. Okay? Does anybody know what TB means? Tuberculosis. Okay, tuberculosis comes from a bacteria called Mycobacterium tuberculosis. Right? Mycobacterium tuberculosis, or tuberculosis, having tuberculosis, is a acid-fast type of bacteria. Okay? Now, since it has a waxy layer, it is really tough to kill. A lot of antibiotics don't work too well with tuberculosis.
Is that in your bloodstream?
Yeah, it can be in your bloodstream, and then it ends up being in your respiratory system. Yeah, that's what it primarily affects. But yes, it can go through the bloodstream. Because of which, since it's a large waxy layer, it doesn't And on top of that, the immune system doesn't do too well as far as getting through that waxy layer. So it's like a big, big, huge barrier. But the disadvantage that the bacteria has is that it doesn't grow very fast. And if you know about tuberculosis, it is a slow grower, which means somebody that has tuberculosis untreated, it will take years and years and years for them to finally pass away. Okay? They used to call this back in the 1800s consumption. If you ever heard this word consumption back in the, if you ever study this in history or look at the Westerns and somebody died of consumption or that person has consumption, that's tuberculosis. What we mean by consumption means that whatever they got sick with is consuming their body. So eventually over time, they get sicker and more pale and more thinner and more weaker and those types of things. That's consumption. So here is a acid-fast bacteria. Now notice, kind of lied to you, but it does have a peptidoglycan layer, but on top of it is the mycolic acid. That's that thick, waxy layer. So gram staining doesn't do anything. It doesn't. The stain doesn't get past this point. Does that make sense? It doesn't get past this point. It just bounces off because it's lipids, so it just bounces right off. So if you did gram staining here, it wouldn't do anything. You go through all the thing, it wouldn't do anything. But because of the, wouldn't get to the underlying thing. Here's the red appearance, right? Red, pink appearance that you see, and there's Mycobacterium tuberculosis, right? That's TB, right? And so if somebody had TB, for example, they would probably take some sputum, which means that's that lower kind of mucus stuff that you can hack up from your lower part of your lungs. You would take that and run a sample on it, and acid fast stain, and you would see that come through, right? Which is diagnosed. That's for tuberculosis.
So is it kind of like pneumonia almost, or?
Yeah, it resembles a lot of pneumonia in the fact that, there's a lot of respiratory distress and there is a lot of coughing, hacking to try to get that stuff out. That's your body's response to trying to get it, the mucus that's kind of settling there. And that the fluid is settling in there. So there's going to be some edema in the lungs, right? So some fluid adding to the lungs. Yeah, so very much like pneumonia. Yeah, very similar. Okay, mycoplasma. Again, there's no cell wall per se. Usually they're going to harbor inside other cells. That's how they live, basically. Inside other cells, other living cells. And tuberculosis does this. We call them a tubercle, which means you'll see in a cell little kind of like areas where the bacteria is growing inside. Kind of little tiny area. And that's what you see there. And then they're paleomorphic, right? So they mean they have a different shape. They have very different shapes. Go to this part, right? All right, so when we talk about substances going in and out of a cell, we call this transporting or moving or diffusing, basically, stuff back and forth. Because as I mentioned, when we're talking about a cell wall, we know that, for example, the plasma membrane is a passive barrier, right? It only lets certain things back and forth. Now, living organisms have two major ways of transporting then things that they need in the cell or things that they need to take out of the cell out. The two ways or the two types of transport is passive versus active transport. In the fact that the passive does not require energy to fulfill the movement of stuff back and forth. But the active does. So the key word for active is it does require energy. Energy is always going to be used in the form of ATP. And when we break ATP and break off a phosphate group, what we release is one phosphate, but that comes with the energy. And the energy is liberated, and that energy can be put to work like transporting big things back and forth. Okay? That's a channel protein. That's a channel protein. Does that make sense? It's facilitating. It's like a gate. It's the gate. It's a gate opener. Opens the gate. Here, come on in, and closes it. All right? So let's talk about the process of osmosis. Whenever we talk about the process of osmosis, we are talking about the solution and where it goes. Not the solutes, but the solution. And where does the solution go? And in a living organism, the thing that we talk about, the key word to think about osmosis is the movement of this type of solution. And that is what? Water, right? As I told you, that water is, made up the environment, cells, water. So in osmosis, we're looking at the behavior of water being a solution and how it behaves with a certain amount of solutes. Okay? Osmosis is also known as the diffusion of water, by the way. That's what we see. So this can be confusing for some students, right? Some students have really struggled through osmosis, right, to understand how this works, right? But we know for a fact, because water's what water is chemically and how it behaves and everything, that water can move readily from the inside of the cell to the outside of the cell with little channels that open up. Or sometimes it can go through the cell wall. It just takes a while, over a long time, but it will go through, and not in great amounts, but it will, right? And so water will react depending on how many solids there are on the inside of the cell versus outside the cell. Okay? Water will always move, here's the key word, water will always move to the area, if it's inside or outside, of the highest concentration of solutes. Wherever that is, water will move. Will move. In an effort to balance out the concentration level or dilute the concentration. So whatever area has the higher solute concentration, water will move there. Is that going into the cell? Sometimes. Is that going out of the cell? Sometimes. Depends on solute concentration on the inside and the outside cell. Okay. In a hypertonic environment, hypertonic solution, we are talking about the solutions that the cell is bathed in, the outside. If it's hypertonic, that means there's a lot of solutes on the outside of the cell, more so than the inside of the cell. There's more on the outside than there are on the inside. If you'll notice in this example here, we notice that there's many more purple dots or solutes on the outside of the cell than there are on the inside. I don't have to count them to know I can see there's more purple on the outside, right? I mean, you just look at it, you already know. So where is water going to move? To the area of the higher concentration. So if water's on the inside of the cell, guess where it's moving? Out. So it will move out, just like the arrows indicate. When it does that, we see a change in the yellow line. What is the yellow line, folks? Can everybody guess what that is or know what that is?
Is that the plasma membrane?
Yeah, somebody else said it. Plasma membrane, plasma membrane, right? What about this other line that's right here, this gray brownish line?
With the cell wall.
Cell wall. The cell wall doesn't change. It's like a fence. It doesn't really move. But what does move is the phospholipids, or the phospholipid bilayer, right? So because water goes out, it's very much like a water balloon. right? You know what a water balloon is? Fill up with water like this, right? You don't tie it off, but let's say you let the water go. What's going to happen to the balloon? Okay, so that behaves exactly like the phospholipid bilayer would behave. So if you take more water out, it's going to shrink and shrink and shrink. That's why they call this plasmolysis. It's plasma lysis. Out. Okay? In a hypotonic environment, that means there's less solutes on the outside of the cell than on the inside. And you can see that definitely is true, right? There's more purple dots on the inside of this cell than on the outside, obviously. You can count them, but we already know that just by looking at it. Where is water going to move? Towards the higher concentrated solutes, and eventually everything's going to move in now, right? And just like adding water to a water balloon, when you add more water, it's going to blow up, right? But the cell wall doesn't move. So when that plasma membrane blows up to the cell wall, it's going to gain more pressure because water's just flooding in. Okay? And eventually, if you leave it unchecked, what will happen to the cell wall and the plasma membrane? It'll break open. It'll pop like the balloon will pop. Spill everything out. Everything will go out like this. And you have cell death, because that after you break open like that, I mean, there's no going back. Cannot repair it. Cell's dead. It will die. The last one, the isotonic. Yes, ma'am. Go ahead.
Oh, I thought that the barrier on the outside really didn't like water like coming in. I thought it was only a little bit that would come in, and that the cell wouldn't notice.
Well, so what's going to occur is that the channels will open up.
Mm-hmm.
So they will allow the water to go through, and in the peptidoglycan cell wall layer, water can pass through between the chemical bonds. Water can go past that point, but in the plasma membrane, the channels will open up. Water channels will open up. Yeah, because water will automatically want to push its way through, and the channels will open up. Yeah. In the isotonic environment, there's an equal number of solutes on the inside versus the outside. So water flows both ways. This way and this way. Both ways. Okay? Okay, so let's talk about active transport. Active transport requires energy, right? Moving stuff back and forth. Sometimes we can do this because we can go against the concentration gradient. So it's like going up or upriver. You know, the river flows this way, right? And it's like swimming upriver, right? You require energy to do that, and in fact, active transport can pass stuff against the concentration gradient that normally wants to go the other way. Here, okay, by adding a phosphate group to the glucose, we call this glucose 6-phosphate, or glucose 6P equals glucose 6-phosphate. But by doing this, it allows the molecule to continue to come through. Sometimes that occurs. Those are the three main ways that. So flagella. Flagella are used for movement or motility, right? That's what that word means, motility, movement. Okay? Some organisms have one flagella, some of them have two, some of them have more than that. Depends on their species. And we're going to look at the names for how many flagella they have. Flagella are quite popular, so about half of the bacteria have flagella, the other half don't. So what are flagella, right? First of all, they're made up of a protein called flagellin. That's the protein that it's made up of. And it's a really long, thin protein called this a filament that sticks out and kind of runs outside of the cell, like a tail, right? Now you can use the word tail to help you memorize that a flagella is like a tail, but please do not say it is a tail, because it's not a tail. They don't have a tail in that regard. Because a tail in biology, when you talk about an animal having a tail, has a total different function than a flagella does. Okay.
So this is more, the flagella works more like, not like legs, but to help movement.
Yeah, it's more of like in a boat, a propeller, where the propeller will spin and then move the boat, right? In water. We use a boat because this bacteria will be in a water-based solution, right? Or this, right? This is my little drawing of my sperm. One little sperm, right? But you know that even eukaryotic organisms like humans, we have cells that have flagella like sperm do. And the sperm and its tail, right, or its flagella, actually, move like this to allow this portion to move, right, up the female, right, to the uterus to hopefully interact with what? Fertilized egg, or an egg to fertilize it, right? Because the tail doesn't matter. What's inside is the DNA, right? Once that DNA builds with the, mature egg in the fallopian tube, it will get fertilized and then implant into the uterus, right? But the reason why I'm mentioning sperm is because that takes that trip, helps the sperm take the trip all the way up. Well, in the same regard, these help move the in its environment. So this is your typical flagella. There's one there. And we're looking at chemically how it's attached to what? The cell wall and the plasma membrane. Okay? And in the gram-positive bacteria, since there's just a peptidoglycan cell wall and the plasma membrane, it's attached that way. And in the gram-negative, it has to go through that plasma membrane, that outer membrane, through the cell wall, and then into the plasma membrane. So we have these rings, but there's proteins that kind of hold it in place. Here's all those flagellins. So if you look really close in your book, you can see that they're made up of these little dots kind of bind it together. And it's a bunch of those proteins binded together, right? The bacteria actually make this in here and then push it out of the cell, and then that becomes the tail or flagella. Now here's an interesting, neat way this actually works for those bacteria that have flagella. We call this the run and tumble system, and we've actually looked at this in research. It's really cool to look at. You can actually look this up. Scientists, researchers have actually discovered this and said, okay, bacteria actually move in this pattern, right? Runs and tumbles. In the fact that they will put all their flagella on one end and start moving them, and the bacteria will move or run. So we call that run, moving. And then at a certain point, they'll stop and kind of open all their flagella and float in the air, or float in the water, like this, right, for a while. And then they just decide, okay, let me put all my flagella together and start moving, and I run again and tumble and run and tumble, run and tumble. And what they're trying to do is get to some kind of stimulus. It senses in the solution nutrients or a cell or chemicals, basically, and it's going to go towards that area. That's how it knows to move. It doesn't have eyes and have ears. But that's how bacteria do this. Through a series of runs and tumbles. Now, there's not a set number of how many runs and tumbles or anything like that. We just know it runs and tumbles. That makes sense? Now, why do you think it does this? Run, tumble, run, tumble, run, tumble. Why can't it just go from, like, let's say here and move straight there? Why does it have to run and tumble? Why can't it just, like a boat, just take off and then finally get there? What do you mean by refueling?
Well, whenever it stops, it's like, I guess, getting more energy to be able to move it.
Is it because it just can't see it, it has to sense it, and so it goes one direction? Is it?
Well, both are true, actually. Both are true. One is very true, right? Where in the fact that they're using a bunch of ATP molecules, and they use them up. Because they use them up, they run out of fuel. Then they have to gather and break down because of metabolism and make more ATP, and then they use more ATP, number one. And number two, they have to reposition where they're going to eventually go. So they can't, minor adjustments, they can't do that. So what they do is they run to that area, they know it's that area, so they'll go that way. And then they'll stop to readjust, reread, and say, okay, how far am I from there? And then keep going and keep going and going. So both are actually true. Talking about that, right? Chemicals. They can respond to chemicals, stimulation, right? They can also respond to light. Some of them need light, energy, right? Light has energy. Some of them can also respond to oxygen levels. Because remember I told you that there's, some of them need oxygen, right, to make ATP. Remember this on the board? It's just left up here. Glad I did, right? Some of them need oxygen. We call this aerobic. Some of them don't need oxygen. That's anaerobic, right? So they can respond to oxygen levels. So they can actually go away from oxygen if they don't like it, or they can go toward oxygen. These are just the three names we use for that. Okay, what about the arrangement, if you will, of flagella? Here are the four major types of arrangements, or how many flagella that the bacteria can have. So we can classify bacteria based on how many flagella they have. Monotricious, Loptotricious, Amphitricious, Paratricious. Might need some practice, pronouncing those, but I think in the back of that book they have a pronunciation kind of table back there. But Monotricious, Loptotricious, Amphitricious, Paratricious. Talk about if you have one flagella, like this one down here. If you have a couple of bacteria, a couple of flagella on one end of the bacteria, Loptotricious. If you're Amphitricious, you have them on both ends, like you see there. And Paratricious, means that you have them everywhere, all over your cell. Sometimes flagella look like this, and especially for the spirochetes. Remember the spirochetes have a spiral shaped, they kind of look like, I told you, they look like worms. Some folks that look at them under the microscope think they're worms, but they're actually bacteria. And they have this corkscrew shape, and inside of the cell wall, it's in yellow, we've got a bunch of long flagella that kind of encircle. And that's what you can see right here in this X, right? This little long, we're just a cut is a cross cut, and we're looking at the side view. They're in between the plasma membrane and the cell wall, and then what they do is they actually rotate. Those proteins rotate. When they rotate, they move the whole cell wall around, and when they move the whole cell around, the corkscrew kind of works. What are fimbriae? Short proteins, bristle, they're not flagella, but what they do is help stick to surfaces or stick to other bacteria. These are the ones that are really important when we study biofilms. We talked about biofilms, right? How they kind of form this community against the surface, and I told you that the teeth form biofilms, and over time that can actually be damaging to the catheters, and food prepping surfaces like cutting boards and those kind of things, right? They're short, they're bristle-like, they're not fimbriae. So these are not, I'm sorry, they're not flagella. These are fimbriae right here. And they act very much like Velcro. You know how Velcro works, right? Kind of sticks to one surface like this and then doesn't move. That's kind of what these do. We see them very commonly in gram-negative bacteria. These pili will transfer bacteria or some genetic information from one bacteria to another. Now, if this genetic bacteria, this genetic information of one bacteria codes for, let's say, how we can become more resistant to an antibiotic, going back to your example, and can transfer from one bacteria to another, it will do so, right? So you can make more things more resistant, other bacteria more resistant. We do this through a process called sex pili, through conjugation, okay? Which we'll revisit again later, okay? Here's another type of feature that you have outside of a cell. We call this the glycocalyx. It's a carbohydrate kind of, production that organisms will undergo, right? And they secrete this, and it's filled up with water and carbohydrates, so it's very sticky. It kind of is like mucus, but it isn't mucus, okay? So don't say this is mucus. It's not, but it's very mucusy, okay? And the fact that it's sticky, it helps stick bacteria to things like tissue, surfaces. Protects bacteria from desiccation. What does desiccation mean? Does anybody know what this is?
It's like decompose, like breaking down.
No. Good guess, but it's not. Desiccation, not defecation, because that's a total different thing. Defecation is pooping. Desiccation is something different. Desiccation is drying out or dehydrating. Does that make sense? Desiccate. To desiccate means to dehydrate, dry out. So it protects it from drying out. Can protect it from, let's say, disinfections or types of chemicals or antibiotics. So there's two types of glycocalyx, a slime layer and a capsule. Difference is the slime layer is very unorganized, loosely packed, a lot of water, sticky. The capsule is more organized, tightly packed, forms like a shell almost, both of which are sticky, but they different, right? So look at the slime layer down here. Everything in green is the slime layer. I like how they dyed it green, like Slimer and Ghostbusters. Right? But that's kind of what it is, that little loose kind of, it's loose, right? It's out there, right? Sticks really well. Capsules are like this, right? So the white portion, in fact, all this from the red line in all the way to the bacteria, which is the rod right there, all that white area is all the capsule. We've got a special way to dye or stain capsules, and that's what the white area is just showing you that that's all capsule. Okay, so inside the cell of a prokaryote, we have cytoplasm, right? Now the word cytoplasm is a big word to describe everything inside of a cell of a prokaryote and a eukaryote, right? So this is the fluid, these are the enzymes, these are some of the organelles in there, and some of the other things that we call inclusions. Inclusions are little storage areas inside of the bacteria or prokaryote that hold certain things. They store extra molecules, substances that the bacteria may use for its daily activity. What's also found in the cytoplasm is a area where the DNA is kind of packed together. We call that the nucleoid region, or the nucleoid. This is not the nucleus, because remember, a prokaryote doesn't have a nucleus, but it does have DNA, and that DNA is found in a certain area in the bacteria. We would call that area nucleoid.
Is that the only place where DNA is stored? Within the cell?
That's the only place that this type of genetic material is stored. This type of DNA is stored. But there's other places that you store genetic material in a bacteria. Call that a plasmid. We'll talk about that also. Now, does it necessarily need to be right in the middle like we see here? No. Sometimes the nucleoid is found in one side of the bacteria or another. So it doesn't necessarily need to be right in the center like it is here. The nucleoid is just telling you where is the DNA. That's where the DNA is collected. Now, most of the DNA is found here. There are other areas where there's portions of DNA that are found in another place of the bacteria. Now, one of the organelles that's really important for all living organisms and that prokaryotes have is a ribosome. Now, if you know this from general biology, right? Ribosomes are responsible for making proteins, right? They assemble the amino acids and they bind them together, making a protein. And we know that proteins get used for everything in a cell, right? Enzymes, proteins on the cell, on the plasma membrane, the peptidal glycan, the peptides get made by the ribosome, all that stuff. Now, they have a 70S ribosome. Prokaryotes have a 70S ribosome. Now, the S represents the size of the ribosome. You don't need to know the measurement, but you can actually look up what the S means. And it's a unit of measurement for the number of genetic material it has and the number of proteins it's made up of. But it's the key word is 70S for prokaryotes. What do eukaryotes have, by the way? Because we haven't got to eukaryotes, nor I don't think we will right now. 80S. There's a bigger ribosome in the eukaryotic organisms than the prokaryote, right? And we talk about these subunits, okay? Large and small subunits. Because in fact, a ribosome is put together like you see here. You see that there? You see the two sides kind of put together? A ribosome lives like this. It exists like this inside the cytoplasm. It's only when it reads genetic material that it opens up like this and then closes on the genetic material and reads the genetic material. The two openings, one and the other, are called the 50S and the 30S subunit. Okay? 50 and 30S. So there's some numbers to remember, right? Prokaryote, 70S ribosome complete, made up of a large subunit of 50 and a smaller subunit of 30.
Wait, but doesn't that add up to 80?
Yes, does add up to 80, but that's not what you need to do. You cannot calculate it because it's totally different. Okay? The measurement is totally different. So you got to make sure that you take your mind off of adding the numbers. Just know that the whole thing together is 70. And the two subunits, one is 50 and one is 30. I don't want to go into, because there's a whole other discussion that will waste time because it doesn't really matter to you unless you get into genetics. Like if you're going later on in your career in genetics, then that would mean something important. But for your purpose, it's not necessary. Yeah, there's so much science that, there's so much detail, so it's lovely to talk about all of it. Okay, so what I'm going to do here, it's 9:40. We're going to stop right here, okay? What I do want you to do is continue on from where I left off, right? So you're responsible for finishing off this part, okay? You're also, what's also needed to be covered by you guys is chapter 4, right? That's the eukaryotic organisms. We didn't get to that part, unfortunately, which I thought we weren't going to get to that, so that's just what it is. Okay, so